Good afternoon, everybody. I'm Petr Kluson. I'm Vice President of the European Federation of Chemical Engineering, and I'm very glad that we are starting the autumn series of Spotlight Talks with the one devoted to food. I'd like to express my thanks to all members of the EFCE section on food for organizing this. We are really grateful. We support this activity. Here we have Kemal Aganovic as secretary of the food section of EFCE. He will introduce the section, or he may talk briefly about the section and about the spotlights today. We have 4 speakers on interesting topics. I'm sure that all of us, we will enjoy it. Maybe I will warm up the EFCE soup at the moment. Martin, may I ask you kindly, Martin Pusch is the head of the Scientific Secretariat of European Federation of Chemical Engineering. So Martin, may I ask you for sharing the invitation for the new forum. We organize quite a lot of events throughout the year. Of course, the most common, the most known is, the best known is the European Congress on Chemical Engineering, but I'd like to draw your attention on the nearest one, if not talking about spotlights. 5th of December this year, we are actually opening or starting activities of a new section of the European Federation within the European Forum on New Technologies, and the new section is called Chemical Engineering as applied to medicine. We all believe that it is something which is very, very important, that chemical engineering slightly moves from those standard issues and topics also towards a lot of the biochemistry, as we in the past evidenced for biotechnologies, but now it goes straight forward to medicine. And so there was a kind of pressure on starting activities of this new section. So all of you are cordially invited to join us. It's one-day event with very low registration and also on an attractive place in Paris, the Advent time. So all of you are cordially welcome. So thank you for this screen. And I think that right now it's time for Kemal for introducing our afternoon program. Thank you once more, Kemal, for doing this, and it's your turn now.
Thank you, Petr, and for the introduction, and thank you, Martin, also for the opportunity to have some activities under the umbrella of EFCE. As Petr already mentioned, I'm a secretary of the EFCE section on food, which is active and better known perhaps for the European PhD workshop on food engineering and technology that is organized yearly, every year for the best PhD students in Europe and European universities and research institutions. However, in the last period of time, and in particular during the pandemic, we learned that webinars are also very good tools for networking, for disseminating results, and reaching out to different audiences. Today we selected a topic on the proteins, and in particular plant proteins. The importance of plant proteins today is out of different aspects don't have to be emphasized, I guess. Most of you are involved in food processing or food development, involving plant proteins, and importance of these proteins and their functionality in final food products is of crucial importance, not only for the right processing condition and processing technology, but also to meet the consumer's expectation, and thus contribute to maybe an optimal and a more sustainable food system. If you're working with plant proteins, probably you already faced some challenges. If not, I'm sure that our excellent speakers today will try to bring this topic a little bit more forward to you. We have 4 excellent speakers from the industry, from academia, from the research part, and I'm looking forward to hear their talks Please keep in mind that we will keep the lectures or the presentations up to 25 minutes and allow 5 minutes of questions and discussions in the end for the audience. I ask kindly to write questions for that foreseen section in a Zoom for the questions and answers, or in a chat. We will find them either way, and we will ask them at the end of the session. So I'm happy to welcome our first speaker for today's webinar on the plant proteins, which is Avi Shpigelman from Technion, associate professor at Technion for already some years from the Faculty of Biotechnology and Food Engineering, where he also did his master's in the biotechnology and food engineering and PhD in the biotechnology. Avi is also very active in the EFCE section, food, and works a lot with the new technologies and potential application of proteins and potential possibilities for functionalization of plant proteins using different approaches. So with that having said, Avi, I can say the screen is yours.
Okay, do you see well my presentation?
Not yet.
Not yet, no.
Not yet. Okay. Screen. Now?
Yes, now I see it.
Okay. Okay, thank you very much. Thank you, Kemal, for inviting me. Thank you, EFCE, for supporting those spotlight talks. And today it will be my my pleasure to give like a short presentation about what we have been doing regarding plant protein sources and technologies, especially pressure-based technologies, and also give some overview about some challenges that the whole field faces when we discuss plant-based protein sources. But actually I will start with saying thank you for the people that are doing actually the hard work in the research. So this is my team, and also Anna, that is from Kemal's team. And I do it in the beginning because then in 20 minutes Kemal will tell me to stop talking, and I want to make sure to recognize the people doing the hard work. Okay, so why are we even interested in alternative proteins? Why it is probably one of the hottest topics in food science and in food technology? Well, there are many reasons. Many of them are related to sustainability, to the environmental costs of growing animals for the production of protein. But there are also not a small number of well-known facts related to health aspects, correlating the consumption of plant-based protein and foods rich in plant-based proteins with various chronic diseases. While it's not likely that those are related directly to the protein source itself, but to the whole composition of foods made from animal sources. And there are different concepts related to alternative protein productions. One of them is the cultivation of meat or from stem cells using bioreactors, which is a very interesting field, but a little bit down the road, let's say. There are additional fields related to the utilization of insects as protein sources. Also, a very promising field in terms of the quality of protein and the content of protein. but in terms of the consumer, it's not easy for the Western consumer to accept the fact that he or she should eat insects in the nutrition. There are also proteins developed by fermentation. Precise fermentation is a hot topic, but currently one of the most studied and actually utilized alternative protein sources are proteins that are originating from plants. Actually, we are consuming such products from the start of human nutrition, and we know how to process them to relatively known products, but we are still limited in the possibility to produce from those plant protein sources products that resemble the products from animal sources. And we have a range of plant-based proteins, starting from proteins from cereals, actually a major source of proteins in the human diet, from wheat, from corn, from oats, from rice, and so on and so on. Proteins from legumes like soy and peas and chickpeas, which are actually now very common ingredients in different food products and becoming even more common. Proteins from oilseeds, like sunflower, flax seeds, and there is now a growing interest in rapeseed and in pumpkin seeds. In sesame seeds, there is a large fraction of proteins from nuts, proteins from edible seeds, even from tubers, and we will see some examples of the utilization of tuber proteins in the presentation. So it all looks perfectly perfect, and we have a range of sources of proteins without the utilization of animals, but in an excellent paper review that I suggest that anyone who is interested in this field, you should read it, by the group of Rafael Mazenga from ETH Zurich. He presented the challenges, summarized well the challenges related to the utilization of plant-based proteins. Of course, there are nutritional challenges related to different and unbalanced amino acid composition or the presence of anti-nutritional factors like phytate and digestibility, and issues related to taste. There are very often undesirable flavors in those protein sources. But when we are speaking about food engineering, we are very often specifically interested in the technical functionality of those proteins. The structural features of the plant-based proteins are very different from the structural features from animal sources. They do not actually, in most cases, resemble the fibrous structure expected in meat, but they are also do not actually well resemble the structure expected in products like milk. So we have poor aqueous solubility. We have quite a significant complex structure. We have a variety of proteins in each plant, a different response to external factors like temperature, and we will discuss pressure later on. They have, in most cases, poorer emulsification and foaming and gelation and texturizing ability compared to the similar or to what we expect in animal-based proteins. And this faces a challenge for a simple replacement, let's say, of animal-based proteins in our formulation with plant-based proteins. Of course, in terms of meat, and this will not be the topic of today's talk, we have several strategies for the formation of texture from plant-based proteins. The most common and most well studied and discussed is the extrusion, but in addition to the extrusion, we also have utilization of proteins from fungi. We have wet spinning, we have free structuring, we have shear cell technology, electrospinning, there are different ways, both bottom-up and top-down strategies for structuring meat analogs. And of course, extrusion is the most common one, but again, this is not the topic of today's lecture. So we will talk about how and if we can use hydrostatic and hydrodynamic pressure to manipulate plant protein technofunctionality, trying to at least partially overcome the technofunctionality hurdles of plant-based proteins. So just a very brief intro about the two different concepts I have suggested, the hydrostatic and hydrodynamic pressure. So the hydrostatic pressure is a technology used in the industry by the application of high levels of pressures, where pressures of up to 600 megapascals, approximately equivalent of having 3 elephants on a surface area of a quarter. And those are usually applied for the microbial inactivation in packed foods. And we also have the dynamic high pressure technology, the high pressure homogenization, which uses pressures a little bit lower, up to 400 megapascals. But this is a continuous technology capable of destructing droplets and forming emulsions. And it is actually a partially thermal technology. When we very briefly compare those two technologies, so in hydrostatic high pressure, we can say that covalent bonds are practically unaffected, but there is a strong, significant effect by pressure on the ionic, hydrogen, and hydrophobic interactions, and this results in protein structure manipulation, which of course is very important for the topic of today's discussions. This also means that enzymes' activity can be either enhanced or inhibited. We have the isostatic rule, meaning that pressure is transferred to the whole system immediately, and importantly, we have pH changes due to the increased ionization. In high pressure homogenization, we see that this type of technology, with the combination of pressure and the resulting cavitation and shear stress, we see particle size reduction, polymer degradation, extractability, and it also allows us to have some microbial inactivation. But again, this is not the focus of today's talk. So, what will we talk about? So, the first thing that I want to show is a study we performed about pressure-based gelation of potato protein isolate. So we wish to see, can we form a hydrogel using pressure in comparison to heat, and how will the functionality and the texture of those gels be different in the comparison between heat and pressure. And we also wanted to present the potential advantage of the utilization of pressure in compare to thermogelation. So let's start with some really simplistic information about the spectral changes occurring due to the application of pressure and due to the application of temperature. And on the top left, you can see that with increase at pH 3, with increased pressures at the temperature of 40 degrees C, which is below the denaturation temperature of potato proteins, and this is the maximum temperature reached during high pressure processing in the conditions we chose to work with, we could see a mild increase in the absorbance, possibly related to the fact that we see some structural changes in the protein at those conditions. On the other hand, at pH 7, we saw a very significant increase in the absorbance, and later on we will discuss why this happens. On the other hand, at temperature, at those conditions with increasing temperature, we practically didn't see any changes due to heating, except of a small decrease in absorbance, likely related to better solubilization of the protein. Okay, so after performing this test, we actually performed a fermentation. We performed pressure treatment and temperature treatment of those solutions at a higher concentration, at 10% of potato protein isolate, and we could see that at different conditions we can get rheology that presents the rheology of physical gels. At pressure conditions, we obtained physical gels, clear physical gels at pH 3, and without forming strong physical gels at pH 7. On the other hand, by heating, we could clearly see the formation of physical gels starting from 50 degrees C, and even starting to see some initial beginning of gelation at 45 degrees C. Potentially also important is that when the maximal temperature in the pressure transmitting fluid did not surpass 30 degrees C, we were not able to get pressure stable gel at any of those conditions. So we can conclude that pressure can result in the formation of pressure-induced gelation, but the outcome is different compared to the gelation at temperature. If we look at this interesting point regarding the combined effect of temperature, this was also mentioned in a study performed about 24-25 years ago, showing that, for example, for egg yolk, we could only see an increase in hardness, meaning the formation of gels, when the initial temperature was above 0 degrees C. While for a valbumin, it was a more complex behavior, but we also saw the effect of temperature in addition to the effect of pressure. And here, where we believe also comes in play the effect of pressure on pH. So it is quite well known that pressure can affect the pH in buffered systems and also in food systems, and even in pure protein solution, as was seen here for a beta-lactoglobulin solution, showing a decrease in pH with increasing pressure. But the measurement of pH under pressure is quite complex, and there are no commercial units capable of measuring pH under pressure, and very often we see different methods that are not direct methods of measuring the pH. But if we consider the fact that in most cases, and also if we consider the calculation that at the buffer used for pH 7, we would expect acidification, bringing the protein solution closer to the pI of the potato proteins, we could suggest that at pH 7, the application of pressure resulted in the formation of aggregates due to the rapid decrease in pH, and this rapid decrease in pH did not allow, and the aggregation did not allow the formation of the 3D network as happened by heating or by pressure at pH 3. What about the textural properties? So if we look, for example, at the TPA, the texture profile analysis of both thermal and pressure gels, we could see differences, again, indicating of different structures of the gel. The hardness of the gel increased with increasing pressure. It increased also with increasing temperature, but there were significant differences in the hardness between the maximal pressure used and the maximal temperature used for the studies. Differences were also observed in the adhesiveness, and also the water holding capacity was quite different, with the maximum water holding capacity actually presented in the temperature showing in the lowest temperature showing gelation. We could also see the fact that the most significant contributor to the structural formation of gels, both by heating and by pressure, were actually the high molecular weight proteins of the isolate and not the patatin, although the patatin was also involved in the 3D network. Okay, so what could be an advantage for the utilization of pressure-based gelation instead of thermal-based gelation? Well, for example, it could be the entrapment of compounds that are thermally liable. In this case, we used vitamin C, a well-known thermally liable compound, as a model, and we measured the remaining concentration of vitamin C in gels showing similar textural profile, meaning gels formed at 60 degrees C and gels formed at 500 megapascals. And of course, we could easily see that the relative concentration of ascorbic acid was significantly higher for the gels formed at 500 megapascals compared to gels formed at 60 degrees C due to the negative effect of heating. The next important step in the studies and the utilization of pressure for resolving some of the hurdles of plant-based proteins would be to study their digestibility. And those are not my works, but I find them quite important to mention. Those are the works of Professor Carmen Moraro from Cornell, showing that, as expected, the trypsin inhibitory activity is actually not inactivated by the application of pressure. It was almost like in the untreated one, while the heat treatment significantly reduced the relative trypsin inhibitory activity both in lentils and in faba bean. But on the other hand, we did not see any negative effect of this residual trypsin inhibitory activity on the in vitro digestibility of those two protein sources. But at the end, it would be important and interesting to find ways, maybe by mixing some treatments, to increase the digestibility of plant-based proteins, because it is one of the well-known hurdles of the utilization of plant-based proteins. Additional interesting effect could be use high pressure as a pre-treatment before thermal gelation. And it was shown that for several sources that if we pre-treat plant proteins by pressure, later on thermal gelation could result in better functionality, for example, a stronger gel strength. On the other hand, it will be significantly affected by the protein source and by the external conditions like pH that I mentioned before, because we would not like to result in complete aggregation by pressurization before the thermal gelation that we want to use to form the gel. Another example I would like to present is to show you the effect of pressure versus temperature on the spectral properties of a spirulina extract. We hypothesize that pressure will negatively impact the color of phycocyanins due to the protein structural changes, but we believe that it can potentially allow better optimization in comparison to thermal treatment. Pigments from spirulina are well-known pigments in providing blue color in the food industry, but they are known to be very liable to thermal treatment. And this is partially due to the fact that the pigment is bound to the protein and structural changes in this complex results in the loss of the blue color that we wish to conserve. And when we studied the phycobilly proteins that we have extracted from spirulina, we saw indeed that heat treatment results in significant changes in the absorbance, but pressure also results in changes, although those are different changes in comparison to the thermal treatment. When we studied the kinetics of those changes, we could quantify the change in absorbance at 620 nanometers, showing that both pressure and temperature result in the degradation and color loss, but it seems that the rates can allow some optimization of the degradation. We also wish to see, is it just the loss of protein structure that is responsible for the loss of color? And for that, we have extracted the pigment itself and the similar compound purified and compared the changes again by pressure and by temperature, showing that it is likely that the protein structural changes, both due to pressure and due to temperatures, are the ones responsible for the processing induced color loss that we actually wish to avoid. Again, we believe that optimization of the processing condition will allow find conditions that will allow, on the one hand, microbial inactivation, and on the other, significant preservation of the blue color of the pigment. And the last example, and I will really briefly show it, is the utilization of high pressure homogenization as a tool to stabilize potato protein emulsions, allowing LAB, lactic acid bacteria fermentation, and allowing us to form plant-based yogurt alternatives. So the general process involves the mixing of sugar as a carbohydrate source for the fermentation, the protein source, solubilizing the protein by high pressure homogenization, meaning improving the physical stability of the proteins, adding the oil, premixing them, high pressure homogenizing them again, and thus allowing to form an emulsion that is stable enough to have a fermentation that resulting in a homogeneous fermented protein structure. So the first outcome would be the outcome of HPH on solubility of the protein itself without the oil. And we could see, while the potato protein is a relatively stable protein source, but we still see sediment, for example, as you can see here on the top. But by application of high pressure homogenization, the sediment is significantly smaller and we receive a more homogeneous protein solution. When we look at the color of the solution after a high pressure homogenization at different pressures is whiter, and of course the particle size are smaller. If we look at the conditions also after adding the oil, we could see that the emulsion made by high pressure homogenization using oil and the high pressure homogenized potato protein isolate was significantly more stable in terms of creaming velocity in comparison to the unhomogenized solution. And this increase in stability can allow us to perform fermentation. And the fermentation was monitored when we saw the expected increase in lactic acid bacteria during the fermentation, similar to what happens in milk. We also saw the decrease in pH, again, similar to what happens in milk, and thus allowing us to obtain gels formed by the fermentation from the emulsions, and the gels made by high pressure homogenization were significantly more stable against physical separation, as was studied by Lumicizer. We also performed those studies at different content of oil, and in all conditions, the application of high pressure homogenization, especially high pressure homogenization at high pressures of 200 megapascals, resulted in significantly better hardness of the gel and whiter emulsion and more stable yogurt-like alternative. And to summarize some take-home messages, we can say that hydrostatic high pressure is a potential tool for engineering plant-based protein-rich products and those technologies can be used beyond the microbial inactivation they are known to perform. And I would also like to emphasize that to better understand the potential for techno-functional improvement or more comprehensive, we need a more comprehensive understanding that includes an understanding of the source, the method of extraction, the initial properties of the protein. Is it in aggregated state or not? We need to keep in mind the kinetics. We need to keep in mind that temperature can affect also under pressure, together with pressure. And we can also say that pressure can be beneficial for color preservations in systems containing pigment protein complexes, and that, for example, high pressure homogenization is a powerful processing and pre-processing tool for attenuating plant protein functionality. And with that, I would like to thank you for listening. And I don't know if I have time for questions, but I'm open for questions.
Thank you, Avi, for the nice overview. There's no much time for the questions. Let's see if there are any in the chat appearing or in the section for questions.
I'm here if anyone wants to ask.
So far, there are no questions, and for the sake of time, I gave you a little bit of more time to tell the story till the end. I would suggest that we move to the next speaker. If there are some questions appearing during the second talk, we'll keep them in mind and we'll answer accordingly. With having said that, thank you, Avi. I'm happy to announce our second speaker of today's webinar, Xiaoai Guo from DIL, from German Institute of Food Technologies, will tell us about a relatively novel protein source, maybe also not that novel. It was already quite significantly investigated in 50s and 60s, namely protein from green biomass like leaves and grass. Xiaoai is a mechanical process engineer with a degree, a master degree and PhD degree from the Brandenburg University of Technology and Cottbus, and has been since 3 years involved in the research in the Department of Structure and Functionality at DIL. So Xiaoai, please tell us a little bit about possibilities for a new protein source.
Okay, thank you for your introduction. Now let's move to share. Can you see my presentation?
Yes, we see it. It's good.
Okay, so yeah, so today I would like to talk a little bit about our research work here about the extraction of proteins from green plants and show you some selected functional properties. So here at first, I would like to give a brief introduction and the motivation, and then I would like to talk about the extraction and the fractionation of proteins from green plants, including raw juice pressing and the extraction process and the fractionation process. And after that, I show you some selected results and potential applications. And at the last, I would like to summarize my talk. So some days ago, I read some information from the internet. Yeah, here it deals about the global green plant-based proteins market. So here we can clearly see this tendency. Yeah, it increases every year. So we can see the potential market. There are also many researchers and also many industries deal with green plant-based protein. So in our talk, I would like to briefly introduce the extraction of plant-based protein from green biomasses here. For example, from different kinds of green plants, lucina, grass, and also some kind of mixtures from different plants, like a blend of grass and clover. And then we try to develop a novel extraction process technology for an upscaling and optimization, stabilization, and separation process and drying process. So after that, we can achieve some alternative plant proteins and to achieve some functional properties for potential applications. For example, these white proteins have good functional properties, maybe are very interesting for human food, and the green proteins are also interesting for animal feed and some other potential industrial applications. So to further improve the extraction efficiency, we can also use some additional physical and enzymatic technologies. We can combine this with juicing process. For example, we can use ultrasound, pulsed electric field, and the microwave or high pressure and enzymes. So we study this protein extraction in our laboratory. We can use a lab scale, juice pressure, and we can separate the precipitate proteins with a laboratory centrifuge. And we also study the protein extraction based on technical pilot scale. So yeah. So here we can see this setup are much larger than the laboratory centrifuge. The protein extraction from green plants, basically we focus on the soluble protein fractions. For example, here we can see the protein extraction from green leaves. At first, the first step, we should press the leaves or the plants. We get the raw juicy, and after that, we can do the next step, protein precipitation. So here there are different kinds of methods that we can use. For example, we can change the pH value and also use heating coagulation by improving the temperature and also the fermentation. And we also did some experiment with flocculants. And some other methods could be used like direct freezing, drying, and ultrafiltration. So yeah, here just show how we can do that. So the first step, we can harvest the plants in the field or on the farm. And after that, we can transport the harvest plant to the press facility. And yeah, and there we can place and a raw juice. And before we do the protein extraction, we can filter some plant debris, some large particles. Yeah. So this flowchart just to show you how we extract the proteins from the mixture ryegrass and white clover. So the first fractioning harvest grass, and here just now I mentioned sometimes we can combine some additional physical techniques. Here we use PEF system, PEF system. This just to increase the yield and of the plant juicy, because this basically helps to broken the cell membrane, and it helps to to help to get more juicy from the plant cells. So after pressing and sieving, we can get the raw juicy. So here we do the separation to remove the plant cell debris. So after that, we can do the precipitation using purified juicing, by adjust the pH value and heating up to 60 degrees. After the precipitation, we can separate the protein. So the separation step 2. After separation, we get the sediments. This is our achieved as precipitated proteins. So after that, we can do the drying. So basically in our experiment, we use the freezing, drying. After that, we get the protein powder. So yeah, here just shows the pilot pressing facility combined with a PEF system. So basically here the feeding system and pass through the PEF system and transport to the screw press system. After that, we can connect the raw juicy and the press the cake of the or the press the plant can be removed. So on the right-hand side, we can see the separation system. So here we use a continuous disc separator to achieve the precipitated proteins. So here we can do, in our technical center, we can continuous separation, do continuous separation of about 500 or more than 500 liter. Okay, so yeah, these two figures just to we can do a comparison of the juicy yields by using and without using the PEF treatment system. So here we can clearly see that by using the PEF system, we can increase the juicy yield by about 25% and the crude protein about 30%. So this table to evaluate or to compare this system, we connect the samples at different process steps. For example, on the left-hand side, we can see we take the samples of the raw juice and the purified juice and the plant cell debris, and after precipitation and the separation, we take the sample of suppletant and the precipitated protein or sedimental. So here in the middle of this table, we can see the crude proteins at each step. So here finally the precipitate proteins contained reach about 40 or 37 gram per 100 gram in dry matter. For comparison, we also did the similar extraction without using PEF system. So here we can see that the crude protein contained is lower than that with PEF system. Okay, so at each step, we took the sample and also we did the chemical composition analysis. And so here we can clearly see at each step, we can get different, or the protein or the chemical composition have different values. Yeah, so and here we can clearly see finally, we get the precipitate protein about, just now we see 37 gram per 100 gram in dry matter. So here we can see besides the protein and dietary fiber and saccharide, and also we can see it contains a lot of minerals. It would also be interesting for future food application. And to characterize the minerals, we also do the EDX analysis of the ash from the samples. Yeah, here connected at 5 different process states. So here we can see, and there exists relatively high component or the content of carbon and oxygen, and both are coming from the oxide, and also here we can see that in plant proteins, and there exists a relatively higher potassium and some other minerals. Okay, so and to see or to evaluate the protein and the fat, from the complex structures, here we did the conflux nasal scanning microscope analysis. Here the raw juicy, you can find a lot of compact proteins and fat structures. So after separations and purified juice, you find fewer fat and fat structures. So here we can clearly see separated plant cell debris here. This is the separated complex. So here we see the very complex, complicated structures. So after precipitation in the sedimentary, we can see relatively homogeneous structures of protein and fat. And in the substance, we can find very, very few fat. Okay, so now I would like to give a brief summary to these experiments. So here we just now show you or demonstrate a large-scale processing facility for production of grass juice and protein. And also, by combining an industry-scale PEF system, we can increase the yield of juice and crude protein. And here is the capacity is much higher than the lab scale research. Here we can do 500 or 400 liter juicy. And this separation and precipitation can be done continuously, and the separation can also be done with a disk separator on a pilot scale continuously. So next topic, I would like to show a little bit about the fractionation of plant proteins. Here we use Lucena. And so here, similarly, at the first step, we press the juicy, and after that separation, we get the raw juicy. And after pH value setting, here up to 6.5 after separation to remove some plant debris or some large particles, the superlatant will be used for precipitation or fractionation. So here, basically, we use based on the sorting out effect. So here we use ammonium sulfate. Yeah. So by adding a different amount of ammonium sulfate in solution to our protein solution and mixed, and after the precipitation, we also do the separation to remove or to separate the precipitate and protein fractions. So we can repeat it, and by increasing the volume or the volume ratio of the ammonium sulfate to the protein solution, we can repeat it. So after that, we can get different fractions. Okay, so here after separation, we do the dialysis and drying. So we get different protein fractions on the right-hand side. On the right-hand side, we can clearly see the achieved protein fractions. So here in the first three fractions, we can see some, it looks somewhat green. Yeah, it comes from the chloroplastic proteins, and the next few fractions white proteins, they are mostly rubisco proteins. Okay, so here for precipitation or fractionation, here we did the experiment by using the superlatents at different pH values. So here we can clearly see that the protein solution depends on the pH value. This help us to do the protein fractionation. For example, just now I mentioned for Lucena, and here at pH 6.5, we get relative higher protein solubility. So after precipitation and after dialysis, and we did isoelectric point analysis of each protein fraction. So here we can see the IEP range from 4.5 to 4.7. So here we can also see obvious influence on the functional property. With this achieved protein powders fractions, we started the foaming capacity. So here for comparison, we also use the animal protein. So here we use the egg protein. And so here see, and at the beginning, the first three fractions show relatively lower foam capacity. And here fraction 4, 5, 6, 7 show a better foam capacity. And similarly, we can see a better foam stability for the fraction 4, 5, 6, 7. So here we can see they are comparable to the egg protein. And here we also do or calculate the coefficients. This is a combination or contribution from the foam capacity and the stability, CF divided by K. So here it's visible, it's obvious that the fraction 5 and 6 show better foam properties. And for these 9 fractions, we also do the gel formation, but there is no significant gel formation without functionalization of the protein fraction. So here, we didn't show these results here. So the achieved different protein fractions after crude protein analysis. Here we can see the crude protein contains increases. So here at our first 4 or 5, here we can see increases, and after that, yeah, they have very similar crude protein contents. So, and here the first three fractions have relative no protein content. It may be related due to the green proteins. We also do the sensoric test and the flavor evaluation. Here it shows all these samples, all these fractions have a slightly astringent flavor, as shown, yeah, this red buttons, and the first three fractions show relatively strong grassy flavors than the other ones. Okay, so so here after precipitation, we also do the GPC analysis to see how about the molecular mass of the proteins in each fraction. And so here, yeah, it was summarized in this table. And so here on the left-hand side, we can see for fraction 1, 2, 3, 4, 5, we can find the more proteins or high share of proteins range larger than 117 kilodalton. And for the last four fraction, yeah, there shows a relatively high share of proteins in the small range of the molecules. But what are these small proteins? Yeah, it needs to be further identified. But for these large protein molecules, we find that they are Rubisco protein molecules. Okay, so now I would like to summarize the fractionation of the plant proteins retrieved from the Lucena and Juicy. So just now we can see that we can, by using sorting out effect, we can fractionate different protein molecules or protein fractions that show different colors. Yeah. And these green proteins are very interesting for maybe for animal feed, and the white protein are interesting for future food applications. And also, we found that the fractions 4, 5, 6, 7 show better foaming property and stability, and fraction 5 and 6 have a better foaming properties that it would be interesting for food foaming applications. Yeah, and also here just now I mentioned for the gel formation, we didn't find the significant gel formation. So the next step maybe we'll functionalize the precipitate protein fractions to see how to improve the gel behavior. And also during the precipitation, the techno-functional properties are also related or are influenced by the protein mass fractions and also protein composition. Okay, like the formation of complexes between protein and reactive carbohydrates. So, and these questions, yeah, are still, yeah, to be explored or to be studied in the future. So, and that's for my talk. And I would like to thank you very much for your attention, especially we would like to express our thanks to Christoph Dyer for his kind of providing the green plants to press grass juicy on his farm, and also to our dear Greenland project research teams, Dr. Kemal yeah, Aganovic and Ulrich Andreas and Christian, Edward, and Johannes, and also Dr. Volker, as well as some other colleagues for their kind of technical support during the experiment, and also for the sample analysis and for some useful discussion. And yeah, as well as to the financial support from the Greenland research project. So yeah, that's all from my side, and I would like to answer your questions.
Thank you, Joey, for a nice overview on, well, relatively new protein source, considering the needs for the proteins and the growing population. There is no doubt that we will need also new protein sources beside the existing ones that were mentioned by Avi, for example, in the first talk. And the utilization of a green biomass and the green land is definitely one of worth looking into, considering the abundance of that protein. Are there any questions from the audience in the chat or in the section on research and questions and answers? I don't see any questions so far. Maybe I will go with the one. Did you try to check the different, how the seasonality affects the protein functionality? If you, for example, harvest the alfalfa or whatever green biomass you're working with, I assume that you would see differences if it is harvested in June and in November. Are there any, do we have any data on that?
Yes, because here I didn't include it in these slides. And in our past experiments, we also see the fiber change and also the fractionated protein content changes with or are related to the harvest time or season. Yeah. For example, in the September, we get relatively lower protein content from the rice protein fractions. Yeah. So we are planning to do the research by using the new harvest Lucena in November or at the beginning of December, so we can compare the harvest season in this year. Yeah.
Okay.
So
yeah, advantage
basically overview and on the protein fractionation. And for the functionality, the new experiments, we are doing this analysis this week. It's not finished, so we are looking forward to these new results. Yeah.
There is one question in the chat section from Knut Olaf Stradkvern. If in the case of Lucerne protein, did the chlorophyll separate from protein by increasing the ammonium sulfate concentration?
So here the green proteins, we estimate they are chloroplastic proteins. They are also soluble proteins. Yeah. So here in our experiment, we can see that at the beginning, we got the green proteins because at the beginning we used relatively low concentration of the ammonium sulfate.
So it is with the increasing ammonium sulfate concentration, the green color gets less.
Yeah, that's less.
So we can assume that chlorophyll separation from protein actually takes place with increasing concentration of ammonium sulfate.
Yeah, will increase. Yeah, will content, because in the last 5 or 6 fractions we didn't find green proteins. Yeah, they are white. Yeah.
Okay, there's one last question, Joy. Is there any effect of the temperature along with the during this precipitation of the protein on the functional properties of isolated proteins. So far, I remember you tried to keep the temperature below where negative thermal effect might occur in the denaturation. Is that right?
Yes, at the moment, the temperature are lower than the denaturation temperature.
What was the temperature of precipitation?
Precipitation here we use at 60 degree in
60
this
degrees.
experiment. Yeah.
Good for most of
We
the
can
plants.
we also did some experiment like 65 or 70 degree. Yeah, we also did the experiment on.
But I assume this was the
laboratory
best temperature and
yeah,
pH
it's the best
combination
temperature.
for
Yeah.
precipitation.
Yeah. Because this
That
was
is
we
still
did
below.
last year. Yeah. So this year we are trying to do at other order under other conditions or temperature or pH values. Yeah.
I guess for plant proteins, everything that goes lower than 75, a bit less, is not that harmful for the functionality of protein, I would assume.
Yeah. So for thermal heating and coagulation, basically they are, or the temperature is between 16 to 85, something like that in this temperature range. Yeah.
Okay, thank you, Joy, very much. We have to move to another speaker, although they are definitely interesting discussions. Our next speaker is...
Thank you.
Thank you, Joy. Our next speaker is Volker Lammers. Volker Lammers is also coming from DIL. He's the head of the department Process Engineering, focusing mostly on different extrusion technologies. Volker Lammers is a process engineer with master degree in TU Munich and PhD from ETH Zurich, where he did also his PhD on extrusion technologies. So Volker, very welcome, and tell us a little bit about extrusion.
Thank you, Kemal, for the introduction, and thanks to EFCE for the invitation. I hope everyone can see my screen. Kemal, maybe can you give a okay?
It's okay.
It's okay. Thank you very much. Oh, so I started already here at the end. That's of course not correct. So I start at the first slide. That's better. So I will give you. Sorry. I mixed something up. It was the wrong one.
Yeah,
So,
just change it. Yeah.
Yeah.
Perfect.
So, okay, sorry for that. So I will also report a little bit about plant proteins, but more about the fractionation of plant proteins and production of concentrates suitable for extrusion applications to produce meat analog products or related products. So what was the motivation? It's more or less having said before in the talks by Avi and also by Joy. We see a growing world population, climate crisis, resource scarcity, and other issues. And so the rising demand for functional plant proteins, to serve that, we need to work on new technologies and developing new solutions for that growing market. The majority of plant proteins, as we know them and as we use them today, are based on wet concentration and isolation procedures. So a grain or plant raw material is being milled and then processed through various wet procedures using a lot of amounts of water, energy, and chemicals to get functional property or isolate or protein isolate with targeted properties at the end. So this requires a lot of resources, and how can we reduce that amount? So we look and investigate dry separation processes based on different technologies. So what is known is separation based on different particle size or density, so air classification, sieving, etc. But this has a limited efficiency and limited separation yield. So we look into others as well, namely triboelectric separation, which will be the main focus of today's talk. And based on the example of oil press cakes and as underutilized proteins, which are rich in or side streams that can be used better than it is as of today. So we have the goal to implement an environmentally friendly dry separation process and to test it based on rapeseed protein and lupin protein. And then at the end, we look on the applicability of the enriched fractions in extrusion technology. So how does the triboelectric separation work? It's based on the triboelectric effect. So particles are charged by particle-particle interactions and particle wall interactions. So it's basically a property of the particle surface, and depending on the surface chemistry, the electric properties and particle morphology, some particles tend more to load positive and others to be negative, some others more to be neutral. And depending on these surface properties, we can also involve a separation of such protein, such particles. And this process, known as triboelectric separation, is already commercially used in the non-food areas, mainly in mineral processing of fly ash, coal, and some others, and as well in plastics and waste recycling. So our goal is to implement this technology in the food and feed sector to produce functional protein concentrates by this resource-friendly technology. How does the triboelectric separation work? We are working here at DIL with a belt separation system, and it is designed as follows. We have a chamber where we have a top positive electrode and a bottom negative electrode, and within this chamber we have a belt, a perforated belt, which is moving like this. And in this feed port, the particle, the feed material, is conveyed into this chamber. And by the electrodes, some particles tend more to go to the positive electrode, so to the one side, and some others who are more affine to the negative electrode go into the other direction. So we get at the end two fractions. Of course, they are not 100% positive and negative. We always have a mixture of fractions, but we are getting to an enrichment of that. So the advantages of that technology is that it's water-free. We don't need any chemicals, relatively low energy requirements, and we preserve the initial functionality of the proteins and raw materials. So the belt separation system we are using is designed by our partner STET, Separation Technology and Equipment from the US. They have provided a separating system here in our pilot plant, where you can see here the feeding system at the center of the separator and two bags basically at the end of the chambers, of the separation chambers, where the material is then collected, either protein rich or protein poor material. And in this chamber we have a belt moving, and what are the parameters we can adjust? It's the voltage, it's the gap width of the electrodes, the belt speed can be adjusted, and also the feed rate, so the amount of material we put into the separator has an impact, as it is related to the particle-particle interactions. So we have created an experimental design for that. So as said at the beginning, we worked with de-oiled lupin flakes and rapeseed press cake, which was cold pressed, and the initial protein contents were 38% and 34% respectively. And first of all, both were pin milled, so by an impact mill, to further have a size reduction of the material, which is important, I will show later on, for the disintegration of protein and starch bodies. Then materials are put into the belt separator, and you get these two fractions. Here it's called fiber enriched and protein enriched fractions. And those fractions were then further investigated and looked into particle size, color, particle morphology, protein content, mass yields, and color. So you already see here the first results from the feed material, here on the left for lupin and on the right for rapeseed, separated into two fractions, protein-rich and fiber-rich fractions. So what did we learn? Basically, we know that there are crucial factors influencing the separation efficiency and the yield. So it's on the one hand the particle size. So small particles are required as we have a better disintegration of the protein. So protein bodies need to be released, as seen here in the scanning electron microscopy image on the bottom left. So only when we have, through the milling, disintegrated starch and protein, we can also separate them. If they are still sticking together, it's difficult to get a good separation. So if we have already a higher initial protein content, we've seen that it's also beneficial to improve the separation. So separation efficiency also depends on the size of the protein bodies, and the similar effect was observed for both for lupin and for rapeseed press cake. So here we see some size distribution, cumulative size distributions on the upper right, and impact. So the lupin flakes we've milled 3 times, because we've seen after the first time it was still too large. After the second time it was smaller, but no good separation yield. And so we've seen an improvement as smaller as we got it, and at the end I think we were about 20 micron as a mean value here. Yeah. Yeah. Then the second important factor was the composition. So, and here mainly named the lipids. So for the rapeseed press cake, we've observed that there was still high fat content in it, and this relates or resulted in some caking of the electrodes. So it was sticking on the electrodes, and so the efficiency for separation was decreased over time as material was built up, and we've seen some agglomeration of the particles. And it was better when the raw material was further de-oiled below a fat content of 5%. And as the lupin flakes were already de-oiled, we've seen better results with the lupin flakes. And here are the results. So what was possible? We've started, so very initially after the raw content was about 36%. And then after milling, we already had some kind of effect of classification, as some dusty, smaller materials, particles were removed. But we've entered the separator with a protein content of 46% and separated it into a protein-rich fraction having a protein content of 59% and a protein poor or fiber-rich fraction of 22%. And what we've seen here, and what is interesting, we've get a mass yield here of 50% on the for the high protein fraction. So when we look into these separation systems, we always consider protein content, but also the mass yield. So this is the important factor later on for scaling this technology. What is the yield we can get? And what we've seen, smaller particles are beneficial if we run the setup at higher voltages, also better results, and higher belt speed and smaller gap supports the separation. For the rapeseed press cake, we've also observed an increase in protein, and I think here it is also illustrated very well. I hope you can see it also on the screen at home in your monitor, that from the feed material we clearly have a separation into a dark and into a light fraction. So the dark spots here are mainly related to the fibers, and on the one it is fiber poor, and hence the protein content is increased. And obviously we can conclude that the fibers have a different tendency to be charged by this process and then to be separated. So but however, no clear correlation between the process parameters and or no significant correlation, I have to say, was observed so far for the rapeseed press cake. So if we look a little bit more into the particle size of and particle morphology, we've seen here some size distributions, density distributions of the 3 times milled lupin flakes and the separated fractions. So the separated fraction, the E1 was the protein enriched one, which is a little bit, or which is smaller than the protein poor sample, but it's also kind of a separation by size, if we would argue, but yeah, closely related to the surface property. And it's a bimodal distribution, which could be related that some particles are not completely disintegrated. For the rapeseed press cake, it's a similar, let's say, trend, but we have slightly larger particles. Also in the E2 fraction, or the protein-rich fraction, we have the smaller particles and more proteins, and the larger ones are more related to hulls, fibers. Yeah. Illustrated a little bit more the particle morphology here in these microscopic images. On the top, we have the lupin samples, and on the bottom from the rapeseed. And we've seen here larger particles in the E2 or in the protein poor fraction, and smaller ones in the lupin fraction, where we have a better disintegration of the material. And similar it is for rapeseed press cake. So what was then the goal? We have were able to produce separated fractions or protein concentrates by this technology. And we wanted to investigate how suitable are these fractions for extrusion processing, namely for low moisture extrusion, also known as TVP or texturized vegetable proteins, and high moisture extrusion, HME process. So these are the two main extrusion operations known or used today for making meat analog products based on plant proteins. And so I stress this again, the majority of plant proteins are used for one or the other process today are produced by wet procedures. So we believe there is a big benefit if we are able to use this, let's say, minimal processed raw materials in extrusion, and there would be of great value for the entire production chain. So the low moisture extruded products, they are characterized by an expanded structure. We have a cellular structure characterized by air bubbles, air inclusions, and the high moisture extrudates. We have here a fibrous structure, longer fibers and layers oriented in one direction. How are the samples made? Low moisture extrusion, we only add about 20% water to the protein. We mix it, we heat it, we compress it, shear it in the extruder, and then we extrude it through a small expansion die to get these type of products as shown in the slide before. For the high moisture extrusion process, same raw materials, but higher protein, higher water content, so different water protein ratio, also slightly different process parameters. And at the end, we have a long cooling die where the material is is structured. So how is the structure mechanism done? So for the low moisture extrusion, we have this expansion at the die. So basically a high viscous protein water mixture exits the die, and when it exits the die, we have a rapid pressure drop and water evaporates as the mix is still over 100 centigrades. And the cellular structure is then solidified by cooling in ambient condition. For the, here is a typo. So it's wet extrusion, of course, with the cooling die. And the protein melt is cooled down from the outside to the inside. We have a laminar flow profile, which is somehow fixed in the die, and so the subsequent solidification layer by layer gives us then the fibrous structure characteristic for such products, as illustrated here in these images. So we made products based on lupin or rapeseed protein mixed, and I have to say that and admit that. So we still used a little bit of pea protein isolate. As we know, this has a very good functionality for this high moisture extrusion process. And so far, the generated concentrates by triboelectric separation do not have this sufficient functionality. But so we started with mixtures thereof. And from the left to the right, the ratio of pea protein isolate was, no, it's the other way. Left a lot of pea, and then on the right, more lupin. So we have a brighter color. The structure was well, and so a positive impact of the lupin. When we look into the rapeseed, yeah, that is a little bit the biggest impact, let's say, or challenge is the color. So it is still too many dots inside, yeah, and not a good, or the structure is okay, but not very appetizing. So structuring okay for both materials, but the best result, which could be also be, let's say, commercialized in some way, is when we had a ratio of 40% lupine and 60% P, giving a nice structure, and also taste is, of course, of big importance. So before I conclude, just an outlook. What are we doing here at DIL? And you've seen here in industrial scale setup that we're having in our institute, and we did not use that for the samples created, because we have just limited quantities available, but that would be then the outlook for the future, to use this setup either for dry extrusion or wet extrusion, where we have, can produce several hundred kilograms of material extrudates per hour. So that would be available, but certainly one of the next steps. This is how it looks like. And so I come to the conclusion that triboelectric separation is a potential technology for making customized dry fractionated protein concentrates. Particle size and composition have a strong impact on the separation efficiency, as well as the separator design also. I didn't mention that in detail, but the separator design and the process parameters. And we need a sufficient disintegration of protein bodies from the other materials to achieve a good separation and a good separation yield. And ideally, the fat content is as low as possible. So for the future, we will investigate also different milling types and look into the particle, more looking into particle size and particle charge. Then investigating protein functionality before and after separation, and functionality of different fractions, and combining the triboelectric separation with other dry separation technologies such as air classification, sieving, and others, to further improve yield or increase yield. And yeah, the work was mainly done within the framework of two EIT food projects, namely Taste to Meat and Tribotech. So here we collaborated with some partners from University Helsinki, VTT Finland, TU Munich, Napiclarin Biotech, a startup from Poland, Matis Institute in Iceland, and Prolupine, a startup in Germany, working on lupins. And not illustrated here, also DSM and ABP Foodtech were also involved. With that, thank you very much. And if you have questions, I'm happy to answer.
Thank you, Volker, for your contribution on relatively new technology in the powder or powder separation, in particular protein separation. It doesn't seem that we have any further question unanswered, or are there any? I have one or two questions related to the triboelectrical separation. The first one, you mentioned that the fat content is actually not really beneficial for the separation, which is understandable considering the charging of the molecules or particle as a major phenomenon. Is there a maximum level or a lower level where you would say, okay, we can work up to that point?
Yeah, so we had some press cakes from other oilseed materials where we had entered or started testing with 20%, which was way too much. So we didn't get a good separation and also not good yields. So if it's, I would say below 10, we could give it a try.
Okay.
And below 5, ideal. So that's.
So even if we have a biomass that has a 20%, we have to somehow bring it down to 10, 15 in order for Tribor to work properly.
Yeah, and then of course also considering how much energy do we put already in the de-oiling and what type of process is it? Just if it's just mechanical de-oiling, that would be from an energy perspective preferred, but then it's not clear if we get below these certain values. So this is then maybe a trade-off and something need to be considered definitely.
But good, again, we are in a selection of the oil separation technologies, what kind of treatments and pre-treatments we are using, what kind of centrifuges and so on and so on. So we have to think a little bit in before coming to the point of triboelectrical separation. There was another question going in the direction, what would be the major advantage compared to the classical air classification that we know?
Yeah, so air classification, the separation principle is particles are separated based on size and density. And as some particles may stick together, or maybe some starch particles have the same density and size as protein bodies. So we do not, this is limited. And now we want to add another separation mechanism to further increase the separation efficiency. And as I said in the last slide, it should not be like triboelectric separation or air classification. So my guess is that a combination of such technologies will give the best impact. And now we are looking, what should we do first? Air classification and then triboelectric, or maybe the other way around. So there are many opportunities to be tested.
It's good, many research questions. The scientists like that always. There was another question. You did this based on the plant proteins, which were grounded in the powder form. Will it be possible to apply the same separation technology to dried green biomass, for example, grinded leaf?
Yeah, so I don't have experience with that. So I think I would need a little bit more information about the starting material and how it is possible to also first mechanically disintegrate the fractions. So if I have just small particles, but it's all looking the same, it's difficult to separate. So I don't know.
That would be the major criteria: particle size, maybe chemical composition.
Yeah, so particle size, we know that we need to mill down. So at least for the oilseeds, cereals, and legumes that we've tested so far, going, let's say, below or around 50 micron. So it's fine milling. Then we see some good results if we are large. So I don't know the particles in the grinded leaf.
But then we have to know the particle size and maybe what is inside, what kind of particles these are. Maybe something worth trying. That was the last question. If there are any further ideas, I actually wanted to say this in the end.
Thank you very much for your time, and let me know if you have any questions.
Thank you, Robert, for nice insights from another perspective, from industry perspective. So far, I didn't see any questions in the audience. Maybe they need a few minutes. But Joy, it seems actually that your green protein is not hopeless. If we have an aeration technology that can adjust the color, then there is a hope. there is something there. Robert, do you see which market is developing the most rapidly? I would say, I mean, in terms of technology, Europe is always somehow behind a little bit of the States and maybe Asian market, which seem to be a little bit more open to innovation and maybe a little bit also more aware of be it healthy food or quality food or minimally processed whatsoever. I'm not sure if that comes to the purchasing power or simply the awareness. Do you have any figures on that and maybe the reasons for why we see this type of behavior?
Yeah, not so much. I think especially when it comes to cultured meat, you definitely see more openness in Asia, especially in Singapore and in Switzerland. Yeah, I think we're a bit ahead of the rest of the EU, but we still, of course, have to follow EU guidelines. But no, I don't have those kind of figures, like especially for plant-based meat, where it is. No, it's a great question.
Yeah. Good, that would be interesting to see which market. I mean, in terms of investments, I would say that definitely good with Singapore. The United States is one of the most developed or advanced ones. You're also right when you mentioned, I read yesterday or a few days ago, that that the shares and everything around the Beyond Meat crashed and went down by 50% or more. But does it mean that the company is dead or the hype is gone, or is rather that the competition doesn't sleep? So we have much more options, not only the Beyond Meat that is doing these types of products, but actually many more.
Yeah, definitely. I mean, only time will tell. We don't know if it's going to crash, or they also just had a big hiring phase last year. So they grew very quickly and then they shrank, but still overall they did grow. So we'll see. It's hard to predict what will happen to their share price, but yeah, there's definitely more competition coming in. Lots of room for innovation. And we can't just say that because one or two companies aren't doing well, that the whole industry is dead and that it's something not to pay attention to anymore.
Well, it's just in a developing stage, as you mentioned, it didn't even reach the peak yet.
Yeah, that's our hope. Yeah.
Good. There is another question from the audience, well, maybe to all of you, which relates to any solution for improving the mouthfeel for these meat analogs and juiciness in the end and chewiness. Unfortunately, Volker is already out, but what are your opinion on that? Is that something that we can achieve by aeration or functionalization of protein or even fat integration?
Yeah, go ahead.
I think one solution will, in this case, will not be enough. So you can do some aeration to improve the color, which also affects our representation of the product. So if the product is not white and you expect chicken, the mouthfeel will not be good, even if it is good. So, but we will also need a solution for the fat and for the juiciness. And at some point in time, the Ministry of Health of each and every country will come and ask, is it good for us nutritionally, those solutions? And then you will need to answer, okay, what do I do with all the stabilizers that we need to have to stabilize the juiciness? Many solutions are still needed, and new problems will for sure arise in the future. I can already see the problems rising, at least in the Israeli Ministry of Health. They do not like those products. They do not say that they are bad, but they might do it in the next year or two.
Good. That's I would say there is a transition phase that also the governments have to go through together with consumers and producers and retailers and everybody that are involved in the production of whatever food that is. There is a generation shift, obviously, and there are different opinions on many things. Actually, I forgot to mention now. Well, yeah, actually, it seems that we have new products, but old problems. I was approached quite a few times if we can use technologies that we use for a meat tenderization to make the plant-based analogs more tender. Could we try, of course, to do these technologies, be it in a mechanical or electrical approach? But it seems that that there is a need and there is a hope for these technologies not to be utilized in a different way.
Yeah, definitely. And there's so much that we can still test, and just an idea could turn into a massively successful startup, as we're seeing. So yeah, if someone has a great idea, there's definitely an incentive to give it a try and see if it might be, a commercially viable solution even.
Sure. All right. I don't see any other further question in the area, nor in the chat. Well, maybe just for the final words in the end, in the conclusion. Obviously, we need a careful selection of raw materials, be it a grass protein or some other protein, but also selection of processes and pre-processes will obviously play a major role in the functionality of plant protein, but also in the final acceptance of these types of products. Of course, on the course of these developments, as Robert mentioned, we have to follow some sustainability aspects, because in the end, the final aim is to get more sustainable, at least from the food and agricultural perspective, or at least to try our best. I once again, the question in the group, would you like to share your slides? I know from Martina, I heard that talks will be recorded, so probably they will be also available to take a look afterwards and maybe to even share them. But if there are any objections in sharing the slides, please complain and object now, or otherwise we assume that you don't have anything against. And with this, I would also like to ask you to send the slides to perhaps to Martina, Petr, and me, so that we then be in the contact with any potential people of interest. I have also prepared the slides with the contact data, which I would like to share with all the participants. If anyone has something against, please raise your hand, and I will delete you from the list. Otherwise, I will just share it for a very few seconds for people to be able to, I hope you can see the slide, to be able to write it down and note a little bit on the data. On my other slide, there's a EFCE team, Petr and Martina, which I will then write you in a very short. So having said that, basically we brought our webinar to the end, and I would like to thank everyone for their participation, and first the audience for being patient and staying late and following our activities. Not to forget also our speakers with their excellent contributions so far. And now I can stop sharing, I assume. And also thanks EFCE team, Petr and Martina, for making this webinar possible, and for a great coordination and putting everything together we provided. In total, these would be my last word. Have a nice evening. If you have to drive somewhere back home, please drive carefully, and hope to see you next time in one of our future EFCE activities, be it the spotlight talks on or some other activities. Thank you and have a nice evening.
Thank you very much.
Thanks so much.